Hello, my name is Sophia N Karagiannis from King's College London. Today I would like to share with you our work on developing IgE class antibodies for cancer immunotherapy, and I will discuss the mechanism by which this antibody class can activate immune cells in the tumor microenvironment. Monoclonal antibody scaffolds designed for cancer therapy can be engineered in a variety of formats, each designed to exert different anti-tumor functions. Fc-mediated effector functions have the potential to activate important immune effector cells, such as macrophages, which are present in significant numbers in the tumor microenvironment, and which potentially can recruit other immune cells, such as monocytes, NK cells, and others into tumors. Effector functions are now increasingly recognized, and efforts to improve these functions could result in the design of better antibody therapies for cancer. Looking at a table of examples of monoclonal antibody treatments which are approved for cancer therapy in the clinic, we can see clearly that all of these antibodies are designed to be with Fc regions of the IgG class. When an effector function activation through the Fc region of immune cells is desired, the antibody is made of the IgG1 class, and whether effector functions are not desired, the antibodies are generally designed with Fc regions of the IgG4 isotype. Again, looking at the indications, one can see that many solid tumors, such as ovarian cancer, do not currently benefit from a range of antibodies therapeutic agents. Importantly, mechanisms of action include both direct effects, such as the inhibition of receptor ligand interactions through the Fab regions of the antibody, but also Fc-mediated functions such as cytotoxicity, ADCC, or phagocytosis, ADCP, or complement activation. It is therefore perhaps desirable to consider how one can influence and improve the effector functions of antibodies for cancer therapy. Tumor-associated immune effector cells, such as macrophages, are often alternatively activated in their phenotype and thus less likely to be activated by therapeutic antibodies. Often these cells express inhibitory Fc receptors, such as Fc gamma R2b, which when engaged by IgG antibodies could trigger negative signals and reduce immune cell activation. B lymphocytes in the tumor microenvironment have also been shown to express inflammatory immunoglobulin isotypes such as IgG4, which are not effective in triggering immune cell activation and may often compete with otherwise activating IgG antibodies for Fc receptor binding and signaling, thereby reducing immune activation in the tumor microenvironment. Different solid tumors such as ovarian cancer, which are characterized by poor prognosis, currently do not benefit from antibody therapeutic agents. Therefore, we set out to ask the question whether we can engineer antibodies that are more likely to be immunoactive in tissues, they're able to operate in Th2 bias conditions, and don't compete with other immunoglobulins for Fc receptor binding. Importantly, we wanted to ask whether it's possible to activate immune cells in the tumor microenvironment which are already there, such as monocytes and macrophages, and which are alternatively activated phenotype. Therefore, we wanted to ask the question whether it's possible that we can engineer an IgE class therapeutic for cancer therapy. And the reason why we consider that IgE may be an interesting antibody class for cancer therapy was because IgE works through a different group of Fc receptors that are not shared with IgG or IgG4, and therefore IgE may not compete with immune blockade mechanisms including IgG4 in the tumor microenvironment. The affinity for IgE for its Fc epsilon receptors on immune cells such as mast cells, macrophages, monocytes, and others, which could be found in the tumor microenvironment, is 100 to 10,000 times higher than IgG1 has for its highest affinity Fc gamma receptor, Fc gamma R1. IgE, unlike IgG, is immunoactive in tissues. It works in Th2 environments. And in fact, although we all know IgE as an antibody that is pathogenic in the context of allergy, IgE antibodies are produced to protect from parasitic infection and can clear large parasites in tissues such as the gut and the skin and work in Th2 inflammatory conditions. Activation of IgE via its Fc receptor can trigger potent inflammatory cytokines, recruitment of important inflammatory cytotoxic cells, and IgE is known to trigger antibody-dependent cell-mediated cytotoxicity and phagocytosis of parasitic cells in tissues. IgE has also been shown to be an important antibody that can trigger antigen presentation and adaptive immunity beyond its own class towards IgG as well. Interestingly, due to the high affinity for IgE for its Fc epsilon receptors, it has the ability to be retained in tissues in the absence of antigen stimulation, and its half-life has been shown in the skin, for example, to be about 2 weeks. However, the half-life of IgG in tissues is 2 to 3 days. The opposite is true in the blood, where IgE has a half-life of 1 or 2 days, whereas IgG has a half-life of 2 to 3 weeks sometimes, which means these antibodies work in different anatomical locations and may be useful for cancer therapy, even complementarily. So IgE can trigger important effector cells in the tumor microenvironment. Macrophages are important effector cells for IgE against parasitic infection clearance. And unlike IgG, IgE does not have an inhibitory receptor. And therefore, there's no clear known break for IgE immune effector activation. We wish to develop an IgE antibody with human IgE Fc regions specific for a tumor-associated antigen in solid tissues. We selected folate receptor alpha as our target for our first-in-class IgE antibody, and this is because we considered this a suitable target for IgE immunotherapy based on its high expression in 70 to 80% of ovarian cancers and in a proportion of basal, breast cancers, mesotheliomas, and other solid tumors. Also because it is expressed in all stages of a disease, and it is considered a marker of poor prognosis in various solid tumors, including ovarian cancer. Its expression in normal tissues is either absent or very restricted, and folate receptor alpha is an emerging target of IgG1 therapeutics. We selected an IgE version of a known IgG1 antibody against folate receptor alpha. The IgG1 version had already been tested in clinical trials. We tested whether an anti-tumor IgE antibody with human Fc regions could restrict the growth of human cancers in immunocompromised mice. For this, we reconstituted the human immune system with PBMCs from donors. In a subcutaneous model of ovarian cancer, we found that IgE, even at half dose of that we gave for IgG, restricted the growth of subcutaneous tumors in these animals. And in a patient-derived model from an ovarian cancer patient, we found that IgE doubled the survival of the mice compared to IgG and other controls. We then designed an immunocompetent syngeneic system in the rat, where we re-engineered our IgE antibody to be of the rat species, and this is because the human IgE Fc receptor and IgE immune system in rats is more similar to humans than the mouse immune system. In this model, in these immunocompetent animals, we introduced the equivalent species rat IgE and IgG antibodies in tumor-bearing animals, and we found that IgE was able to restrict the number of lung tumors as well as the percent of lung occupancy by cancer compared to IgG and control. In the patient-derived model of ovarian carcinoma, human monocytes and macrophages appeared to be recruited in the tumor microenvironment in the remaining tumors in animals that received tumor-reactive IgE. In order to test the hypothesis that monocytes and macrophages may be important effector cells, we repeated these experiments in using human immune cells depleted of monocytes. In the animals which received IgE together with PBMCs, which included monocytes, animals survived for twice as long as controls. When we removed the monocytes from the PBMC populations and treated the animals with IgE, we found that the survival of the animals was the same as controls. However, when we reconstituted are depleted PBMCs with equivalent amount of monocytes. These together with IgE appear to recapitulate the survival advantage we saw with IgE immunotherapy. This gave us the hint that potentially human monocytes and macrophages may be important effector cells in IgE immunotherapy in this context of ovarian cancer. In order to understand whether human monocytes could trigger the death of cancer cells in the presence of anti-tumor IgE, we pre-labeled cancer cells with a green dye, calcium AM, which leaks from cancer cells once they die. And we labeled monocytes in the PBMC population with an anti-CD14 antibody in red. Within 30 minutes of incubation ex vivo of PBMCs with cancer cells in green, one can see the contact between monocytes and cancer cells, green and red. Within 90 minutes, we could see destruction of cancer cells and the beginning of loss of green fluorescence, suggesting that the cancer cells were dying. And we saw remnants of the cancer cells engulfed by monocytes, human monocytes. Within 3 hours, we saw still contact between the tumor cells in green and the cancer cells and the immune cells in red. And we could see that the cancer cells appeared to be dead in contact with immune cells. This was not the case with a non-specific IgE control antibody, where we saw the cancer cells retain their green fluorescence, suggesting that they were viable and often were in contact, but very often not in contact with monocytes. We developed an ADCC assay where we saw that patient-derived immune cells from patients with folate receptor alpha positive and negative ovarian cancer were able to be activated by anti-tumor IgE to kill cancer cells ex vivo. These suggested that monocytes from healthy volunteers and patients with ovarian cancer were able to target and kill cancer cells when triggered by anti-tumor IgE. In the immunocompetent syngeneic rat model of folate receptor alpha expressing cancer, where tumors populated the lungs of animals, we studied the tumor lesions of IgE and IgG-treated animals by immunofluorescence to evaluate the macrophage infiltrate. We found infiltration of rat macrophages in red, surrounding target-expressing folate receptor alpha positive tumor cells in green in all treatment groups. Interestingly, in the remaining lesions of the rats treated with systemic administration of IgE, compared with lesions from rats treated with IgG or vehicle alone, we observed a large infiltration of macrophages surrounding small remaining tumor islands. This strong immune infiltration was also evident in hematoxylin and eosin stained lungs. Transcriptomic analysis of lung tissues from the different treatment groups revealed that IgE immunotherapy was associated with a pro-inflammatory pathway signature. While controls were characterized by cell cycle and survival pathways, IgE treatment was characterized by activation of immune pathways such as NK cell, IL-12, and IL-7, which were not typical of an allergic response, known to be associated with IgE, but more likely of a pro-inflammatory response, more likely to favor anti-tumor immunity. Macrophages isolated from the lungs of rats treated with IgE immunotherapy appear to have a higher expression of TNF-alpha compared to those that received IgG, from animals that received IgG or vehicle control. Macrophages from the IgE-treated animals appear to express higher levels of CD80 as a co-stimulatory molecule and TNF-alpha and IL-10, but they were less likely to express the scavenger receptor CD163, suggesting a pro-inflammatory signature for these macrophages. In the bronchoalveolar lavage fluids of the animals that received different treatments, we found that these from the IgE-treated animals appear to have higher amounts of TNF-alpha and the chemoattractant for macrophages MCP-1. This provided the clues that IgE immunotherapy may polarize macrophages towards a pro-inflammatory and less alternatively activated phenotype, and with secretion of pro-inflammatory mediators and chemoattractants from macrophages in the animal lungs. We then wish to answer the question whether IgE can trigger human macrophages of different types, including those alternatively activated phenotype, which were likely to be found in the tumor microenvironment. We've derived human macrophages into quiescent, classically activated M1-like, and alternately activated M2-like, the latter of which are more likely to be found in tumors. And we found that when we triggered IgE signaling with cross-linking of IgE on the surface of the cells, we found that classical molecules downstream of the Fc epsilon receptors, Fc epsilon 1 and 2, were triggered. This included lin, which is a molecule very directly downstream of Fc epsilon R1 and Fc epsilon R2, and this was clearly found with IgE stimulation of macrophages, especially of the M2 alternately activated phenotype. Additionally, we looked to see whether these macrophage subtypes could be triggered by IgE immunotherapy to kill cancer cells in an antigen-dependent manner. And we found that quiescent macrophages, classically activated and alternatively activated macrophages could kill tumor cells in an antigen-specific fashion more effectively than the equivalent IgG antibody. This suggested that IgE could activate and signal different types of macrophages, quiescent, alternatively and classically activated, to trigger effector functions and to signal immune activation on the downstream of the Fc epsilon receptor pathway. We then wish to understand whether developing an immune complex on the surface of different macrophage subtypes with IgE could influence the pro-inflammatory or anti-inflammatory mediator release by the cells. We are particularly interested in M2 alternatively activated macrophages because these are more likely to be found in the tumor microenvironment. We found that cross-linking IgE on the surface of alternatively activated macrophages particularly trigger the release of pro-inflammatory mediators such as TNFa, MCP-1, interferon gamma, IL-12 and others, but interestingly not the activation and production of IL-4, which is a classic mediator of allergy. This suggested to us that it is possible that IgE immunotherapy can trigger pro-inflammatory activation of alternatively activated macrophages, which is a population of cells that appear to promote tumor growth, but in this case they are being depolarized to secrete pro-inflammatory mediators, which could be triggering an anti-tumor response to the tumor microenvironment. Overall, our data from ex vivo studies of IgE immunotherapy and animal models of IgE treatment in the solid tumor setting suggested that IgE can trigger not only effector functions to kill cancer cells by activating immune cells, but also triggered a pro-inflammatory mediated phenotype, which are associated with the secretion of mediators such as TNF-alpha, MCP-1 and others. These upregulation of MCP-1 brought in more macrophages in the tumor microenvironment, but also the same signature appeared to signal activatory pathways downstream of the Fc epsilon receptor pathway with upregulation of kinases such as lin, fin and others, and associated as well with upregulation of costimulatory molecules such as CD80 and CD40, but also downregulation of scavenger receptors such as 163 and 206. This whole signature, led by TNF-alpha, MCP-1 and others, appeared to be associated with better survival in patients with ovarian cancer, looking at the expression of these combined in the tumor microenvironment. In summary, anti-tumor IgE antibodies have shown anti-tumor effects in several models of cancer, in vitro and in vivo, and in some of the models we have shown better effects compared to the equivalent IgG1 class antibody. In vitro and ex vivo studies have shown that IgE can direct monocytes and macrophages to be recruited into tumors and can trigger effector functions against tumors. And these effects appear to be mediated via a pro-inflammatory mediator axis dominated by cytokines such as TNF-alpha and chemokines such as MCP-1, which is a chemoattractant for macrophages. We believe that IgE immunotherapy can then provide a mechanism by which one can activate these cells against cancer. This work has resulted in an early phase clinical trial of the first-in-class IgE, and it has contributed to the development of the field of allergo-oncology, interested in the links between IgE, allergy, Th2 immunity, and cancer.